Thank you very much. We've heard some impressive talks. And I guess the organizers wanted us to believe that there's still a role for the academician in the development of new gene therapies. And that's the message I'd like to send to you. The last I checked, Bluebird does not own any clinics. And the last I checked, the basic discoveries actually come out of academia that then get moved to the big pharma or even little pharma and get perfected. So the question is, how do you do it if you are in an academic setting in terms of developing gene therapy trials? So I'm going to actually take you through a kind of a case study that has taught me what to do. And so I'm going to follow the idea of the New England Journal, where they have how I treat X, fill in the blank. This is how I do gene therapy trials. So, confronted with the initial issue, your postdoc brings you a discovery, quote unquote. He's actually done some preliminary work and it looks like he has a possible proof of concept that you can treat a disease of interest to you. There are a million questions that arise. And you've got some idea of what the patient population is, who's going to be treated. You even know what who's you have to involve to get going. But ultimately, it's very, very complicated. And I'm going to go through the complexity and try to simplify it for you and show you how we've done it where I work. So the number one point is that of all the things that have to be done, you are not going to be able to do them. You can't do it alone, and that is the number one point to make. You can't even do it with your postdoc, and you can't even do it with your postdoc plus your, if you have a secretary, you can't do it with your secretary. All the regulatory documents, et cetera, et cetera. Don't even try it. I have been through this for many years, and I have tried it that way in the past, and I can confirm you that it doesn't work. So there are some topics I'm going to go through. Assembling the research team, planning ahead, defining the target product, discussing proof of concept issues, and then what meetings do you ask for with the FDA and when? What are the regulatory affairs issues that you could streamline? And then finally, how do you put it all together once approval is there? And you have to then begin the process of bringing patients in and running things efficiently. And so I'm going to talk about what we have in California called the Alpha Clinic concept, which I think is very important as we do these new technologies that are high-tech. They're on patients that are very sick, and they may not be appropriate for the classical clinical research unit in an academic center. So that's the basic area that I'm going to talk about. So let's start about the division of labor. It really is defined by everything before and after your IND and your institutional approvals and other agency approvals. So these boxes are going to get piled up, and you're going to go through the processes, and I'll take you through them in a minute, but you can see the whole issue prior to approvals is a massive one. Just getting ready for the pre-IND and developing a process that you are confident in can yield a product if you're actually going to be producing the product at your center, as we do for many of our trials. And then you're ready to go, and things get even more complicated with recruitment issues, trial initiation issues, scheduling, product release, and coordinating that with the patient schedule and the life issues of the research subject. The treatment then, the biospecimens, the number one deviation when you're trying to complete your trial will be getting the biospecimens to the right labs at the right time with the right name on the, or the right identifying number on the tube. And then, of course, all the data collection. So that's the issue. And the first thing then we're going to do is assemble a team. So I'm going to give you the kind of the autobiography of the zinc finger nuclease trial, which is the first nuclease applied to a stem cell. in a clinical setting. And so we had a group that was very helpful to me. Number one, we had a scientific lead, and that was Phil, who you heard just a minute ago. And he was critical for zinc finger nuclease scientific aspects and how we develop the various approach to the patient and to the change in the stem cells. With him, though, was a very competent project manager, and that was Michael Holmes. And both have gone on to bigger and better things, of course, since that time. And yet that was critical. So you don't have to have the people from your own institution around the table. In fact, what's not shown here, of course, is the conference calls. And conference calls were held once a week for 4 years before we actually got to the point of approvals and ready to start the patients. We had a clinical lead, though, that was expert in transplantation, and we were going to transplant AIDS patients. So we actually went to Amrita Krishnan, who did some of the first transplants in AIDS patients and had the largest experience in this country for such transplants. And she's on our staff, so we were able to elicit her work. We have a David DeGusto in manufacturing, who subsequently has gone on to Stanford, and we now have a person named Angelo Cardoso that has been making these autologous cells with zinc finger nuclease modifications for our patients. And then we have the basic structure for regulations, for financial checkpoints as we go through what we want to do, and we have to live within the reality of the costs. And then, of course, data monitoring and statistics. So all of these people are around the table, and we were meeting once a week. Michael was dutifully scheduling all this and recording it. So as you get different pieces of data and different methodologies, you record them and you write it up, and that is the data bank, let's call it, of text. It will eventually be assembled into various documents that you'll be submitting. But I want to just emphasize this once again. Your postdoc will want to be the project manager, and that's not a good idea. You need a professional. I think you need a scientist. We now have 2 project managers on our projects. Both of them are PhDs in biology or neurosciences or related areas. And although I want to be gender neutral, it is true that women negotiate better than men. And we found that sometimes you can get through log jams very nicely with a female project manager. But they must have overall project vision, communication skills, engagement, motivation, must be able to be competent and have good composure in the face of crisis. Planning ahead is critical, and so, learn how to use a Gantt chart. It's not that easy at first. You need the right software, but it is very important to be milestone driven. And most funding agencies now that you'll be applying to will ask you to have a project manager and ask you to be will require that you are reimbursed based on milestone accomplishments. Okay, so you're sitting around the table for the first time, and you must then define the target product. And that's something that people forget to do. It is the most critical thing at first, because everyone then will understand what it is. You think you have the right vector, shown here, because the preliminary proof of concept was based on that vector. In fact, in our case, it had already been used in T-cell trials that Sangamo had supported at University of Pennsylvania. But the scientific lead may say, hey, we've got something better. Our postdoc has developed a new vector, for example. Let's take that forward. Well, you can say, well, all of our data is based on this one. And so at some point, you have to lock in to the product that you're going to do and the method you're going to do it. Are you going to use some pills for activating the gene, for example? How are you going to infuse the cells? How will they be formulated? You must come to a conclusion on that. So I just thought I'd show you something about that. It's a fluid document. It can change as you learn more things, but it does memorialize where you stand at any given time. And it allows everyone to understand in their particular area of the work what is going to be done. And you can look up how to do this. I'm sure many of you already do it. but the FDA has a draft guidance, and you just have to search FDA TPP. That's a very complex document, though. It's used really for industry. It's pages and pages long. There's a simpler one. The California Institute for Regenerative Medicine has a guidance. Again, you search for CIRM TPP, and you'll come up with a nice little template. And it looks something like this. This is kind of simplified, but there are only a few things that you have to agree on early. The first thing you do is you agree on the name of the product. So as Phil said, it's not the nuclease that is the research product. It's whatever you use the nuclease on. And it's not, or in. It may be, maybe an AAV, for example, or maybe a hematopoietic stem cell in this case. So we called it SB728MR HSPC. It just flows right off the tongue. And yet we knew exactly what it was. It was the Sangamo Biosciences RNA to zinc finger nuclease that attacked CCR5. And we put it into peripheral blood, hematopoietic stem, and progenitor cells. So then the question is, well, what are you going to do with it? What's the eventual indication? If everything goes right, what's the optimal indication? Well, we're going to treat HIV infection. We're going to treat all R5 infections, and we could treat them early on, we could treat them late, whatever. But you'll treat all patients. Well, what's the worst situation? Well, the minimally acceptable would be you only treat the sickest patients who have failed to respond to conventional therapy. Well, then what about biological activity? Well, that was relatively straightforward. We wanted to see CCR5 disruption, and we wanted to see it in the cells that come out of this transplant. And we asked for 70%. That was our blue sky idea. And what would be the worst? Well, we'd like to get at least 40%. We might even go lower now that we know there's some monkey data that shows that lower levels may have an effect on HIV. But that's where we were 4 years ago. What is the endpoint of the product? What's the efficacy endpoint? Well, control of HIV viremia would be when you would actually take patients off. So that means in our clinical trial, we're going to design it so we'll take patients off therapy from antiretrovirals once you reach certain endpoints. And you will control the HIV viremia in our estimation. And what would you accept as safety? Well, you want the related AEs to be less than or equal to a grade 3. You don't want any major genomic problems, such as cancer, which would be a grade 4. And so, but the safety risk, the minimum safety risk would allow a grade 4 or less. Then the dose. Well, the dose was easy because that's established by standard of care in the field. Let's say 5 million cells per kilogram. Not all of them would be genetically modified, but that would be the total reconstituting a dose of stem cells. The minimum would be at least 2 million per cells per kilo. And then how would you formulate it? Well, that was, again, very easy. We were going to freeze these cells. We're going to release them, and then we're going to thaw them and give them as a single cell infusion and a cell suspension. And we'll use standard of care practice for that transplant. So that was it. We established that early on, and now we all know what we're going to be trying to do as we develop everything else. And the next biggest thing, I think, is the proof of concept. So this is the evidence that from an experiment in animals that the potential product or service can be successful, quote unquote. In vitro evidence is already there. You know you can alter the genome of the cells with these mechanisms, and they seem to have an effect in vitro, but it is the animal model that's required. The in vitro evidence is insufficient. And so the question is then, what is the appropriate animal model? And you may have to develop that yourself. In our case, we had Paula Cannon with us, and she was around the table at each of the meetings. And she had developed the NSG mouse for transplantation and then challenged with HIV, and she was able to show that she gets selection of the disrupted CCR5 containing cells that are CD4 cells. And then we took our own product, that is the peripheral blood stem cells. She had used cord blood cells. We went to one step further and showed that you could, in fact, reproduce this in the mouse model. So we thought we had the proof of concept. So then we're ready to go to the FDA. So the question is, do you go with a pre-IND or do you go with a pre-IND? Well, there's a simple rule of thumb. If you have more questions than data, you go with a pre-IND. And if you have more data than questions, you go with a pre-IND. And we went with actually a pre-IND. And it turns out that our initial proposal eventually was changed completely in the 4 years of our process development. We even changed the patient population. We initially had started with a lymphoma population with AIDS, thinking that would be the most ethical approach. But with time, the FDA had allowed healthy HIV positives to have genetically modified cells with chemotherapy. So we switched it. We started with an adenovirus, and we found that that was too toxic. So we changed that. So we actually then, when we came back with our pre-IND, we finally had our act together, and we had a more logical approach. But the goal is to show that the study is safe. We want both the FDA reviewers and our own investigators to say the study is safe to proceed. And the question then is it? And so you ask for a date to discuss this. And it's best to ask for the date first and then send in the packet of information about a month or so later, just before the meeting, a month to 6 weeks later. And that saves you time. Explain in the proposal everything you know about it, the rationale, the product. The product must be defined. If it's a cellular product, what is the cells? What do they look like? What's the histology? What's the karyotype? What's the biochemical markers that are important? What is the genomics or proteomics, if that's important? Show your POC, show your method of manufacturing, your plan for tumorigenicity or toxicity, and then what the clinical schema looks like. And with each of those plans, you ask the agency, does the agency agree with this proposed plan? That is basically, I think, the appropriate stance to make at this meeting, as opposed to, what do you think we should do? That's probably not the question to ask. And then in the mind of the reviewer, this is a very nice paper by a man named Fink, came out in Science, describing what the FDA reviewer is thinking about in terms of whether this is really a safe thing to proceed with. And he's going to be looking at the cellular products that you're making, or the viral vector, the origins of it, perhaps the cell products that derive from this. He wants to know whether they are immunogenic, whether or not they're tumorigenic, of course. And then, of course, when they're in the animal, what's the mechanism of action you think is actually happening? How does it happen? Are there other cells that are involved, et cetera, et cetera? Is there a likelihood of some of these cells becoming malignant? So that's the sort of thing. And you tell the reviewer as much as you know about it, and they will give you some ideas on how to develop better information. You go back to your team after this, and you're ready to really begin the regulatory work. And so here you need, again, a person that can really provide critical attention to detail. You can't have paragraphs that are free-floating and they're wrong, that they've been attached from a previous protocol, say, or a previous even IND. To have the accurate paragraphs, and you cannot tolerate errors in the text. You want a person who's certified in regulatory affairs. In our case, the research teams tend to have a person who then relates to a central office that actually does the IND and relates to the FDA. So it's best in an academic center, I think, to have a single face to the FDA, rather than having 100 PIs that can call up the FDA about their particular product, their particular proposal. And I think you have more efficiency in the long run when your institution has a single, as I call it, face to the FDA. This person must have vision and broad interests and use these skills in multiple other trials, very good communication skills, and cannot be bulldozed by the people around the table who want to move this project faster, but that may subject the project to some other problems. So the integrity and character of the person is critical. So now you're ready to submit to the various review groups at your institution or at other federal agencies. Here you have to have rigor, frequent deadlines, weekly meetings, good communication, and you'll get this done fairly quickly. I'm just going to touch on one thing about this, and that's the RAC review. It changed in the last year, as you probably know. So now there is an attempt to streamline this procedure to limit the number of cases that actually need an NIH Recombinant Advisory Committee review. And so your IRB will determine this, and it's going to say if it's a new vector, never been used before, it's probably a good idea. If it contains preclinical safety data using a model that has never been used before, that may be important. And we may see that with some of these new nuclease-based treatments in the future. And also, if it's something that has possible toxicities that are widely unknown. You're going to inject these iPSCs that have been differentiated into neural stem cells into the brain. And what is the model that you're going to need to actually demonstrate possible toxicities? That probably needs an open review. But now you've got your final approval and you're ready to implement the trial, and you've got all these other things to do. And again, there is many, many things, and we actually have outlined about 50 of these types of things, and I'll show them right here. They basically are in three areas. One is the coordination of the material and the information with the team, the clinical team, the PI and the protocol management team, and there's multiple areas here where you have to get things done. There's coordination with the patient himself. The research subject has a life, and things intervene with your attempted treatments. And I don't have to go into all of these. I'm sure many of you are familiar with this. But you must schedule around this, and you must then schedule the manufacturing to coincide if you have a product that's going to be autologous to bring to the patient. And then all other things happen. The patient has to stay with his dog. Our IRB requires for many of these treatments that have no benefit, quote unquote, that you provide the travel arrangements, you provide the lodging, and you become almost a hotel concierge for many of the problems. We had a patient whose caregiver had a heart attack and went to the ICU in the middle of a treatment. Then you coordinate with things that happen elsewhere around the hospital or around agencies, and you're constantly meeting their requirements as well. So the question is, how can a decent person ever do all this stuff? So this brings us to the final part of this, and this is the so-called concept that was the brainchild of the California Institute for Regenerative Medicine. And I think it's a really good one. It says this, it says, these treatments are different than your father's clinical trials and the trials that are generally done in the very excellent clinical research unit. They involve very sick patients. They involve delivery of cells to your spinal cord injury, to myocardial disease, to retina, to areas that are not generally easily accessible in a research clinic. And they require skills that really require a very good clinical nurse as much as a research nurse. So they set up the Alpha Clinic concept, supported by CIRM, and there are a series of a network of experimental clinics that seek to accelerate stem cell therapeutics and achieve sustainability. And these are being expanded to other parts of the state, we think, and there's a deadline next week for some new applications. But this is what ours looks like. So we actually placed it in a nursing unit called the Day Hospital. It's in a building that is our stem cell production unit. That is where all aphereses are done to support 750 bone marrow transplants. So we had a lot of technology in this building, a lot of freezer farms, and we could bring the cells right up to the third floor and infuse them. And we had excellent nurses there because it was an outpatient bone marrow transplant unit that already was the largest outpatient unit for such stem cell therapies in California. So this is what we did. And it provided a nice interaction between the PI and the protocol management team. We have the concept of what we call the hybrid nurse, who is a nurse that is very qualified to take care of very sick patients as well as the protocol. So overall, it has everything you need. A project manager, it has this coordinator of the nurse. It provides biospecimen coordinators to synchronize the study visits with the actual requirements of the trial. It has regulatory affairs people and data coordinators. And we have a few studies. We have like 20 studies, but they're very complex. Whereas in the institution, there's another 250 therapeutic trials running all the time, but these have then the specialized group. And we have our zinc finger trial here, but we have glioblastoma trials. We have Christine Brown's trial in CAR T cells into the brain, and I'll show you some pictures of that in a minute. We have Sangamo's hemophilia trials and other CAR. So we put all of our complex CAR trials into this unit because the PIs have confidence in I think the nursing skills in that unit. So it has worked out very well. And I just want to refer again to the hybrid nurse. The difference between this and a research nurse is that these nurses report through nursing to senior administration, not through research ops. And that's something that not all institutions can do. In fact, even in the Alpha Clinic network, not all of them do that. But it is very helpful in the long run to get what you need from the clinical care of the patient. It provides guidance to the staff and the patients, and it facilitates the interaction with the other research operations activities. So this shows you an example. This is Christine Brown's study, and this is her colleague, Benem Badi, doing a simultaneous infusion of CAR T cells, and this is the IL-13 receptor alpha 2 cells that were written up in the New England Journal recently. And he's doing, notice he's got a two-handed, he's two-fisted, he's got a syringe in both hands. This is the first patient to be infused simultaneously in 2 sites with CAR T cells, one into the local area of the glioblastoma and one into the ventricle. And then this patient will recover in an hour or 2, 3, and go home. And so this clinic actually has the nursing capability to assess a very sick patient and make sure he can be discharged. And if he has problems, they'll call that nurse up in the clinic and they'll come back. And we give these Alpha Clinics special cards. They're VIPs. If they come in on a weekend and the hospital's very full, they get special attention and they'll find a bed for these patients. So it's actually worked out really nicely. And I want to end there. I want to acknowledge California Institute for Regenerative Medicine for supporting the zinc finger trial that I talked about, but also for this Alpha Clinic concept. We have the group of leadership, and the key person for us is the head of nursing. We actually had the senior vice president for a while, and now we have the associate person. We have a key nurse who's very skilled, and she is the patient care coordinator. We don't pay for all of these nurses, but when they rotate through on a protocol, we pay them by the hour. We have the specimen collection, data collection, regulatory people, and then our key administrative person, and Radhika Stan, who some of you know, is our project manager, and makes all of this stuff work very well. So with that, I will thank you for your attention and entertain questions.
I have a question I'm curious about. So with all these novel therapies, the CAR T cells, the genome-edited cells, are patients pretty excited for these? Are they concerned? Are they, how is it out there?
That's an interesting way to phrase it. I think they are excited. There's certainly, in our trial with AIDS patients, that's part of the cure strategy, and so they're very excited. With the CAR T cells, patients are sick unto death, so they're very excited about the possibility of having a therapy since they failed all of their therapies. So I think that there's a lot of public excitement about this. In California, we're trying to rekindle the public interest in this because of the need to continue this kind of a program that supports stem cell-based therapies. And hopefully that can happen. We'll have to see. Yes?
From your perspective, what are you looking at? What are you looking for in a PhD level person as the postdoc that's bringing you the data versus the person that's the project manager type?
Okay, so the postdoc who's bringing us data has to realize that if the data is IND enhancing, it has to be done especially well and well documented, as opposed to a research paper that may not be as well documented in the laboratory. And we know that many outstanding reports in very outstanding journals cannot be reproduced for some reason. But that is not the case in this. We want to be able to reproduce the data, and we'll document it in a, hopefully in an electronic laboratory record.
Thanks.
Mm-hmm.
Very useful presentation. I appreciate your time this morning on that. My question has to do with, you have the clinical schema in your pre-IND package, and obviously that can change a lot. I'm just curious for your perspective for how mature your clinical scheme was at the point of pre-IND, and did the final clinical scheme look much like what you'd proposed?
For the pre-IND, the scheme was exactly as it went forward. And the tox studies, the tumorigenicity studies that involved extensive work in animals were the same. There's a protocol we proposed in the pre-IND was accepted, and it went into the IND. As I said, the pre-pre, though, was totally different from the pre-IND. And in that 2-year period, 2 to 3 year period, we realized that we hit some stone walls and we had to change our methodology. And so the T cells that had been used with adenovirus we could not use that method, and we had to switch to another method. And it was the Sangamo investigators under Phil and Mike that showed that you could do it with electroporation more efficiently than you could do it with the viral vector. So that's how we got into the electroporation thing. In those days, there was no electroporator with a file at the FDA until Maxite brought theirs to the FDA. So that was the one we had to use, and so we did, and we worked out methods to make that work. Mm-hmm.
Thank you.
That's a great talk. How do you fund it through academia? How do you fund it? I mean, the usual grant mechanisms, foundations.
Okay, so that's a good question. Funding is the problem, obviously, and you have to be able, in our institution, it's a combination of 2 types of funds. You get the grant, and you have already a product that the institution wants to support in some way. It's primarily because it owns IP. So remember, CAR T cells were first developed at City of Hope, so we had IP of interest in that area. So we have a fund that will support those kind of projects. So you link that to your grant funding. So in our case, we had California Institute for Regenerative Medicine funding. Then when it moved to the clinical trial, the CIRM actually had an idea that if it's going to help the company, well, the company should pony up with some money. So they have a strategic partnership in which half of the money comes from the company and half the money comes from the grant to the academic institution. So that has worked out very nicely. And that allows us to run referral clinics in various parts of the country that's funded by the company, Sangamo Therapeutics, and bring them into our central area for treatment. And then we treat them, we get them engrafted, and we send them back to the referral centers, where again, that is run then by the company. So it's a synergy between the academic center and the and companies. And I think that hub and spoke type of structure will work very well for a number of things. Hemophilia is an example. There's hemophilia clinics regionally in this country that are supported by federal and state funds. But they don't have, there's just, let's call them community clinics, essentially. They're a little, I guess, a grade above a community clinic, but they don't have research capabilities in general. So they would go to a central academic center, be treated, and then go back to their center for follow-up. And I think that will be a very good model that companies can follow as well. So they know that their product is being well applied, and in a center, and yet it will be available to the masses through their routine clinics. My opinion. Okay, thank you again.
Right.
Do we have another question?
You answered my question, it's fine. All right, so that'll bring this session to a close. I want to thank all of our speakers, and everybody enjoy the rest of your day. Thank you.